Hello everyone, my name is Natividad and a scientist at Nestle. Today, I will talk about the work we have been doing in terms of understanding the ability of microbial products generated upon fermentation of human milk oligosaccharides, or simply HMO, by infant microbiota to protect against inflammation-induced epithelial barrier dysfunction in vitro. This presentation is part of a larger study in which we tackle the overall impact of HMO on epithelial barrier function. We have a similar webinar presentation that talks about direct effects of HMO on epithelial barrier, and I would highly encourage you to watch it. I would like to preface the presentation by giving a little bit of background on the importance of intestinal epithelial barrier. There is an increasing understanding that a dysregulated or leaky barrier influences the development and outcome of inflammatory diseases as well as allergic and metabolic disorders, both during early stages of life up to adulthood. Early in infancy, the gut epithelial barrier is looser, and it becomes tighter with age. Studies have demonstrated that breastfed infants have faster evolution towards tighter barrier than formula-fed infants. This is associated with higher protection of breastfed babies against inflammatory and metabolic diseases. There are several bioactive molecules present in human milk. Among them are a group of human milk oligosaccharides with different chemical properties. HMOs are fully digested in the small intestine, but are fermented by intestinal microbiota in the colon. The product of microbiota fermentation generates functional metabolites which may have gut epithelial benefits. The direct effects of HMOs on epithelial barrier have been demonstrated previously by other groups. Specifically, intact HMOs are able to modulate the proliferation and differentiation of intestinal epithelial cells. Similarly, as I mentioned in the beginning, we evaluated the direct effect of HMOs, and please feel free to watch the webinar if you're interested to know more about these results. Overall, this gives a reason to believe that HMOs may contribute to the proper development of intestinal barrier in breastfed infants and subsequently protection against diseases later on. Globally, we hypothesized that both intact and HMO fermentation products protect the intestinal epithelial barrier against an inflammatory challenge. Once again, for this presentation, I will concentrate on the HMO fermentation products. We further hypothesized that different HMOs, due to the virtue of their chemical composition, will have differential capacity to impact the barrier. The objective of the study is to determine whether microbial products derived from infant microbiota fermentation of a single HMO or blend of HMOs regulate the epithelial barrier function and its response to inflammatory challenge in vitro. This is clinically irrelevant as intestinal lining is in constant challenge with the environment, whether for infection or food antigens, and having a functional and intact barrier can prevent complications and help in maintenance of intestinal homeostasis and overall health. To address our objectives, we employed an in-vitro model to stimulate the human intestinal microbial ecosystem called SHINE. Briefly, a microbiota from a breastfed baby was cultured and let to be stabilized for 14 days in the system. Then after, the system was fed daily with different treatments. These treatments are 2FL, a long mix of 2FL and LNT, a blend of 6 HMO, and finally a lactose, which serve as our control. Before and 12 days after feeding, the supernatants were collected, centrifuged, and sterile filtered. These supernatants were then used for in vitro barrier assay. Briefly, for this assay, which is shown at the right side of the slide, we use a co-culture of 2 cell lines. One of the cell line is able to produce mucus and the other is an absorptive cell line. The cells were first treated with supernatants for a period of time. We then challenged the system with cytokine TNF-alpha and interferon gamma for additional 48 hours to mimic an inflammatory challenge. Throughout the experiment, we dynamically measured transepithelial resistance, which is measurement of permeability of ions across epithelium. Furthermore, at the end of the experiment, we measured permeability of macromolecules from the apical to the basolateral membrane using a permeability marker called fitsidextrin, which I will refer to as FT for translocation in the following slides. This part represents translocation of food antigens and bacterial products that may induce unnecessary exacerbation of intestinal inflammation. On to the results. We first look at the effect of HMO prior to challenge. Please focus your attention on the figure on the left side of the slides. As demonstrated here, you can see that only the blend of HMO6 significantly increased TER, or transepithelial resistance, compared to control positive, which is a cell group stimulated with inflammatory cytokines. After the challenge, we then measure both transepithelial resistance as well as FT for translocation, and as depicted by the figures on the right side of the slide, the barrier leakiness induced by the inflammatory challenge was significantly decreased in cells treated with HMO, but not lactose control compared to control positive. Overall, the results suggest that products of fermentation of HMO6 is able to protect and reinforce the epithelial barrier in vitro. Similarly, microbial products upon fermentation of 2FL alone, or a combination of 2FL plus LNNT, or a blend of 6-HMO, decrease the deleterious effects of cytokine-induced epithelial barrier dysfunction in vitro. Our results support an important mediating role of microbiota fermentation of HMO on the promotion of gut health and on the protection against intestinal inflammatory disorders by human milk. Thank you for your attention.